Hello everyone. So we have already talked about disaccharides and polysaccharides last time. And what you need to know is the linkage, the monosaccharides and the linkage that used to make up those disaccharides and polysaccharides. So we have talked about two disaccharides, lactose and sucrose, and three polysaccharides, amylose, amylopectin, and glycogen. You also need to know that amylose and amylopectin are the polysaccharides that are used by plants. Glycogen, however, is used by us, by animals and human beings. And also what is the structural difference between those polysaccharides? While amylose is a long chain, amylopectin is the branched molecule but with a few branches. Glycogen is a highly branched molecule. Right, so that's the difference between those three polysaccharides. Okay, so now let's look at question number 30. Starch is composed of amylose and amylopectin. All right, so again, it's used by plants. And when starch is broken down, what sugar is released? Well, it is D-glucose. D-glucose. Okay, we only use D sugars. 31. Glycogen is used by human, all right, to store glucose. We have not only glycogen in muscles, but also in livers. So why would the muscles need a store of glycogen? Well, that's because we can use it immediately. All right, we need immediate use for energy. When we move our muscle cells need the energy. So the glycogen will release glucose, and those glucose will enter the muscle cells and be converted to the molecule that contains energy, which is ATP, and we are going to talk about ATP later. Okay, that is energy current for us. So this is why muscles need a store of glycogen. So why does the liver have a store of glycogen? Well, this is about the function of liver. Well, our liver is a very busy organ. It has a lot of functions. So this is probably this is the second function that we have learned about liver, right? So the first one is about lipids, right? All right, so the function of liver is to store glycogen because we want to maintain the concentration of glucose constant. For example, this morning I had a muffin, and I have a lot of glucose flushed into my system, and my blood sugar level is pretty high, and I want to keep it low. How do I do that? Well, my liver just function to change those glucose into glycogen so that we can decrease the glucose level in blood. And when I'm hungry and my glucose level blood glucose level is low, the liver will release those glucose from glycogen. Therefore, there is a balance, and we use the liver to keep glucose concentration constant. Okay. All right. So question number 32, shown on the previous page, which I show here in the PowerPoint slides. Okay, is the structure of cellulose. Well, cellulose is the fourth polysaccharide that we are going to learn. So it also exists in plants, and the function of cellulose is to keep the structure of plants. so that the plants can be strong, can be stiff, can keep their structure. Okay, so what sugar is found in cellulose? Let's look at this sugar. Well, it is a D-glucose, and the linkage is beta 1,4. It is beta 1,4. Why is it beta? How do we know it's a beta linkage? Well, you look at the linkage and you find the two carbons that's involved in this linkage. One of the carbon is anomeric carbon. You still know what is anomeric carbon, right? So the anomeric carbon is the one that used to be the C double bonded O. And then after we make this ring structure, it becomes COH. That carbon is anomeric carbon. For glucose, it is the first carbon. It is the first carbon, okay? So look at the glucose on the left. It is the first carbon, and the position is beta. And we know it's beta because OH is on top. Okay, top is beta, bottom is alpha, right? All right, so, and we only show the alpha or beta for one of the sugar, but not the other one, because the other one is the fourth carbon, and the fourth carbon is not anomeric carbon. So we only see that the linkage is beta 1,4. Okay, so you still know that the linkage that we find in amylose, amylopectin, and glycogen, they are all alpha. So alpha 1,4 or alpha 1,6, they are all alpha. Can we digest cellulose? That is to see, can we break the linkage of beta? The answer is no, we cannot. We cannot break the linkage of beta, okay, because our enzymes, they do not recognize beta linkage. Well, this makes sense, right? So this is actually why we don't eat grass. All right, grass has a lot of cellulose, and we cannot digest them. It's gonna be, so even if we eat grass, we cannot get a lot of energy from them. But still, you know, cellulose is an important component in our diet, and you know the normal way that we see cellulose, right? It is fibers. So we need fibers for our health, and we just cannot digest it. We cannot get energy from cellulose, but it's still important. All right. Okay, so this is 33. All right, and again, if you have any questions, you should pause and write down your questions and send your questions to my email. Okay, send your questions to my email and I will answer it by the end of the day most of the time. All right, thank you for doing that. So part 3, let's look at how we get energy from sugars, because that's what we need. Okay, this is why we eat things. We want to get energy. 34. So we need to know the meaning of those 3 names. So catabolism, anabolism, metabolism. Catabolism is breakdown of biomolecules. We break it down so that we can get energy. All right. What is anabolism? The other way. Okay, we synthesize those biomolecules. Biosynthesis. We do need to synthesize some biomolecules to make important molecules like proteins, DNA, and those are anabolism. Okay, what is metabolism? Metabolism is to convert food into energy. Okay, that happens when we break down biomolecules. We convert food into energy. Okay, this is metabolism. All right, next question, 35. We're going to talk about first how we convert sugar into energy, okay, because this case is about sugar. All right. So sugars are converted into energy that cells can use, which is ATP, by 3 metabolic pathways. They are glycolysis, citric acid cycle, which is also called, Krebs cycle, TCA, or tricarboxylic acid cycle. Okay, and we are missing the third metabolic pathway, which is ETC. So I add here, and ETC, electron transport chain. Okay, we are going to talk about electron transport chain just in a minute. First, so we are going to go over those three metabolic pathways one by one. So first, let's look at glycolysis. What is glycolysis? It shows here. So if you Google glycolysis, you will find this exact image on the picture here. Okay, it seems very complicated, but we are not going to talk about the details in this process. You don't have to know the enzymes, what happens in those steps. You only need to know the reactant, which is the beginning molecule, which is glucose, and the products. What are the products? Okay, you only need to know those two things. So let's work on those three questions here. 36. What is the final product of glycolysis? What is that? That's the end of glycolysis. It is one of the biologic acids. It's called pyruvate. So, oh wait, I've got 35. Sorry. All right. So what happens in glycolysis? We start from glucose. Each circle means a sugar ring. So means a carbon. Okay. All right. So glucose has 6 carbon, and we start from 6 carbon molecule, and then we end up with 3 carbon molecule, which is pyruvate. And we are not going to lose any carbon because we are making 2 pyruvate. So together we still have 6 carbon. So we start from glucose, which is 6 carbon, and we end up with 2 pyruvate. Together we still have 6 carbon. All right. Okay, so 36, what is the final product of glycolysis? Pyruvate. All right, 2 pyruvates. At the same time, we are going to get 2 ATP and 2 NADH. NADH is pretty important here. We're going to talk about that later. Okay, where are all the carbons from the glucose found? Where do you find the carbon from glucose? You find it in pyruvate. Okay, you find it in pyruvate. They are all in pyruvate. We are not losing it yet. 37. How much ATP is produced during glycolysis? 2 ATP. Okay, that's one of the products of glycolysis. Again, ATP is the energy molecule that we directly use to do any function. For example, we move our muscles, we are thinking, and our brain cells, they need ATP as energy molecule. So they are going to break down ATP to get energy from that. So ATP means energy. 38. NADH is produced during glycolysis. Again, NADH is pretty important. What is the function of NAD plus and NADH? To carry electrons and H plus. To carry electrons and H plus. Hydrogen is super important. We are going to talk about it in electron transport chain, ETC. Okay. All right. So what happens in the first step of TCA cycle? TCA cycle is a pretty complicated pathway. As you can see, it runs as a cycle. So how is it a cycle? Let's look at the beginning molecule, which is on top. It's acetyl-CoA. Acetyl-CoA. That means it has 2 carbon linked with CoA, coenzyme A. All right, CoA is coenzyme A. I have 2 carbon pushed into TCA cycle, and then it becomes citrate, and then it becomes different molecule, and at the end I still have this 2 carbon linked with coenzyme A. six molecule citrate. So after the at the end, I have this 4-carbon oxalic acetate OAA, and then this 4-carbon OAA will combine with acetyl-CoA to make citrate again. So this functions as a cycle. This is why we call it a citric acid cycle. Okay, what happens in the first step of citric acid cycle? I have 2-carbon molecule acetyl-CoA combined with 4-carbon molecule OAA, and then they become a 6-carbon molecule citrate. Okay, what is the job of coenzyme A or CoA? The job of coenzyme A is to carry carbons. Is to carry carbon, 2 carbons. Okay. Next, 41. What happens in the remaining steps of citric acid cycle? Well, again, we are not going to talk about the details here. We only need to know that at the end of the cycle, OAA is regenerated. So we use OAA at the beginning, this 4-carbon molecule at the beginning, and then we generated this OAA at the end of this cycle. Therefore, we can continue this process once we have a new acetyl-CoA coming in. All right, what you need to know here are the products of TCA's pathway. The products are 3-NADH, 2-CO2, 1-GTP, 1-FADH2. GTP is the same as ATP, so you can consider GTP as ATP. So here we do have an energy molecule generated. All right, and we have 3-NADH, 1-FADH2. NADH and FADH2 will be used in ETC in the last process, ETC, electron transport chain, to get more energy, to get more ATP. We're going to talk about that later. Okay, how about CO2? How many CO2 are we generated here? 2 of them. Okay, 2 CO2. How many carbons did we put into this cycle? Acetyl-CoA. Again, acetyl-CoA has 2 carbons. So we are putting in 2 carbon, and then we are releasing 2 CO2. All right, so you can answer question number 42 now, right? Where are all the carbons from glucose found by the end of citric acid cycle? They are all in CO2. They are all in CO2. So 2 CO2 for 1 acetyl-CoA, and the acetyl-CoA is also from glycolysis and originally from glucose. So they will all become CO2. Well, you may wonder that pyruvate only has 3 carbon, but acetyl-CoA has 2 carbon. Where is the third carbon? The third carbon becomes CO2. That's the end of glycolysis. That's actually between glycolysis and the TCA cycle. And are we going to talk about that? We probably will not. So don't worry about that. That carbon will become CO2. All right, so 43, how much ATP is produced in this cycle? Only 1, right? Only 1 ATP or 1 GTP for 1 cycle. Okay. How much NADH? 3 NADH and 1 FADH2. Next, what is the function of FADH2? The same as NADH. The same as NADH, to carry electrons and H+. So again, H+ is pretty important here. Okay, so the function of FADH2 and NADH to carry H+ and electrons. 45. What are the final products of the electron transport chain and oxidative phosphorylation? Alright, so let's talk about ETC here. ETC is a very complicated process. We have 5 complexes here. So you see complex number 1, number 2, number 3, number 4, they are all in the membranes, the membrane of mitochondria. So this happens in mitochondria, the inner membrane of mitochondria. Alright? And the last one, which is the blue one, anyway, So the blue one is called complex number 4. It's also called ATP synthesis. And this giant blue molecule is to produce ATP. How? How does it produce ATP? Complex number 1, 2, 3, 4 will accept electrons from NADH and FADH2. So again, NADH carries and FADH2 carries electrons and H+. And electrons will go through the whole process and then eventually look at where the electrons go. The electrons is like the star, okay, the yellow star. It eventually will go to look at mitochondrial matrix. There is the equation H plus electrons plus oxygen. Okay, so the electron will eventually goes to oxygen and becomes, what is that? Water. Okay, it will become water. Again, oxygen is pretty important here. And I think when we talk about hemoglobin, and the function of hemoglobin is to transport oxygen, and I told you that, we need oxygen to survive, to keep us alive. But where do we need oxygen? Here it is. All right, here it is at the end of ETC process. Oxygen is to accept electrons to become water. Okay, this is why we need oxygen. Without oxygen, the whole process will be stopped and we cannot make ATP and we are dying. All right. So let's look at H+. So we have talked about electrons here, right? And then let's talk about H+. H+ will be pumped from one side of the membrane to the other side. From one side to the other side. Okay. So as you learn from biology, mitochondria has two membranes, the inner membrane and the outer membrane. And these electrons will be pumped from the matrix through the inner membrane to the to the other side. And therefore, I create a gradient of H+, which means that on one side I have a high concentration of H+, which is top here, and the bottom here I have low concentration of H+. So what do you know about those H+? They want to go through a process so that they have a balanced concentration. They have equal concentration between those two sides, right? So where do all those H+ go? They will go through the fifth complex, which is the giant blue molecule. When this H+ goes through this giant molecule, this blue molecule will rotate and it will squeeze ADP and phosphate, squeeze ADP and phosphate, and then make one ATP. So it is totally mechanical. It is totally mechanical. When it rotates, it will change ATP and phosphate to ATP. All right, this is how we generate ATP molecule. And again, ATP is the energy molecule that we need. Okay, so now you can answer question number 45. What are the final products of electron transport chain? Right, we are going to generate ATP. Okay, of course we are going to generate water, but we have a lot of water in our system, so we don't care about water. We do care about ATP. All right, the end product is ATP. All right, so that's the end of this lecture. Bye, guys.